All right. So my name is Seth Brown. This is my second project for molecular biology. I did some research on the polymerase chain reaction. This is my description of it and some of the history about it. So to start, polymerase chain reaction, shortened to PCR usually, is an incredibly important process we use in molecular biology, microbiology, anything that'll study the human body, anything that studies genetic whatsoever. PCR is the standard method to actually determine if a sequence is present. So unfortunately, it's very difficult to actually read a section of DNA. Something that can be done is with PCR, we can take a little segment that we know, or that has a known starting sequence, and then we can make tens of thousands of copies of that one little section. If we can do that, we can determine if that's present, because we can also have our known sequence we want to start with, run our PCR. If we get nothing in the end, it means that sequence wasn't present, it means that was not there. So recently, this is known with COVID-19. Everyone's curious if there's COVID, they can run a PCR reaction to determine whether or not COVID is present. And you do that if there is nothing discovered doing a COVID-19 PCR, COVID-19 was not present, or your experiment was faulty somehow. If you do detect it at the end, you can run a gel, and if you see presence, you see the known sequences there in your gel, okay, this PCR allowed us to see that COVID-19 was present. So the history of it was developed in the 1980s by a couple of gentlemen. There are three main people that worked on it. One doesn't get enough credit. That would be Carl Millis. He didn't receive his Nobel Prize for one of the most important molecular biology discoveries until a decade after he first ran it. One of the major issues with the PCR is your heating and your cooling your DNA. And to do that, or by doing that, you allow your double-stranded helix to separate, and then your primer to connect and actually run your polymerase reaction. The issue with that is to get it hot enough that your DNA will actually denature. Your DNA polymerase molecule will also denature and become useless. So originally you'd have to keep adding a DNA polymerase, a known quantity of DNA polymerase molecules every single time to run the reaction to get it to work, which can be expensive, time-consuming, and not very desirable. But they discovered a type of deep-sea bacteria that lives in thermal vents. It's very hot there, and so their DNA polymerase molecule is way, way more tolerant to these higher temperatures. And we can use that, and it survives those temperatures that'll get the DNA to denature. And so we can keep on using it. We don't have to add new DNA polymerase every single time we want to run one, or every single cycle of running it. The first PCR machine wasn't invented till 4 years after the first PCR was run. It's possible to run it without the machine itself, the thermocycle. However, it's much more difficult, way more labor intensive. Having the machine itself, all you do is set all your samples in there, choose your settings, what temperatures you want to get it to, and you leave and come back later, pull it out, and then you can run your gel to determine how well it worked. So I'm going to be discussing two main types of PCR. We have a traditional PCR, and then I'm also going to go over a real-time PCR. The issue with the traditional PCR is you run it, you know nothing until you go back later, and you can, at that point, run your gel or whatever test you want to do to determine whether your sequence is present, what you want to know about the sequence. The real-time PCR, you know as it's going. You can see if it worked or didn't work mid-project, which is very impressive. So for traditional PCR, you're going to need a couple of tools. You're going to need a template DNA, something you're testing. So for COVID, you'll need the sample from the patient, whether that's from way up in your sinuses where the virus would be present. And there's some processing you'd have to do with that sample before you could run your PCR. But you're going to need to isolate DNA, and then you can start working with it. You're going to need a primer with a known sequence. There's all kinds of ones out there you can buy, and that's just to start your reaction. All right, we want to start with this gene. We know the first couple letters of that gene. We're going to start with that. We can go ahead and get our primer with that sequence. It'll bind, it'll run. We can work from there. And we're going to need a heat-tolerant DNA polymerase, unless you want to keep adding it. Now, Taq DNA polymerase is kind of the standard. You can buy the other stuff, but Taq, or there's a couple other heat-tolerant ones available now. You need all 4 of your deoxy nucleic triphosphates. You're going to need all 4 of your nucleotide bases. That's what those are. You can't be using them if they've been dephosphated already. We're going to need the thermocycle, although it's not absolutely necessary. You can do this with simpler machines than that. First thing we're going to do is denature and anneal. So we have our 2 strands together. We can't read them when they're together. They're stuck there. So we have to separate them. We do that with heat. Get them hot enough, they react enough, they separate and don't stick right back together right away. Then we're going to have to drop our temperature down to 55, 60 degrees. and then our primer will stick. The reason the primer sticks and the DNA doesn't stick right back together is the DNA moves, and just a little bit of misalignment will keep it from sticking back together. But that primer is way smaller, it doesn't get caught on anything, it can stick right easily without any issues like that. Once we get everything set up, we'll bring our temperature up to about 70 degrees. Once we're up there, what we can do is our DNA polymerase will actually start replication. And we'll go ahead and do this. It'll form our DNA. When it gets to the end, it'll just run off. And then we can go ahead and do this again. And again, every time we run it, we're going to double the number of our samples that we're making. Those little short segments that we're going to be making, we're going to be doubling. We can do this over and over and over and over again, make lots and lots of samples. So normally, all right, you've got a 10,000 base pair sequence. You can't measure a 200 base pair sequence on that easily. But if you make 20,000 of that little 100 or 200 base pair sequence, it's way easier to determine if it's present, if it's not present. If you run everything and there's no DNA that you're after, it probably wasn't there in your original sample. The results, we can get lots of copies of that one sequence. I said recently, COVID-19 has been the thing. We've been doing a lot with that. This also gives us the ability to copy a specific gene. We can determine information about that gene both from humans and all kinds of other organisms. It's big in agriculture industry. We're using CRISPR to gene editing software to change the DNA of specific crops we want to grow. We don't use it so much for livestock yet, but that could be an interesting field in the future if you give them bacterial resistance or something, because there's a lot of issues with trying to do that to humans, both ethically and practically, but livestock would be a much easier alternative to that, especially modern research. Next, we're going to go over to real-time PCR, and the advantage of this, like I said, we don't have to run a lab project after the PCR to determine if it worked or didn't. We can see while it's running if it worked or not. So what's the difference? The big one is we're going to be adding other compounds, other molecules to our initial mixture of what we're going to use. And there's two main ones we're going to be adding. And they are the Tachman probe, that is our qPCR indicator, and the Cyber Green. Everything else is basically the same. Might have slightly different settings to accommodate for our indicator molecules, but that's what we'll be working with. Cyber Green is a little simpler, it's a little more economic, but it's somewhat more limited in what you can do with it. The Tachman probe is incredibly useful. There's a lot of different things. You can have different settings for it. Essentially, you can buy different ones that are same over a molecule, but can give you indicators for different things that are happening. So the way they work, the Cyber Green one is very simple. When you have double-stranded DNA, it binds to it and glows. When you denature the DNA, it separates. And then while you're running your actual polymerization, you're creating double-stranded DNA, it'll bind to that again, and so you can observe, we're running double-stranded DNA polymerization, it's starting to glow, we know it's working while it's happening, which is very useful. That allows you to avoid running the entire project, getting to the end and detecting nothing. If this is happening, it means something's being made. It might not be what you want, but something's being made, something is present. It can help diagnose issues while you're running your project, or it can give you your answer right away. The other one is the Tachman probe, but it's a little more complicated. It's built with three main parts. You have a sequence of DNA on it, single-stranded DNA that'll bind to a complementary sequence. We're going to have a quencher molecule, and we're going to have a fluorescent group or fluorescent molecule. And the way these two work is, well, the fluorescent group is near the quencher group. It doesn't do anything. It doesn't light up. It doesn't glow. But when your sequence of DNA will bind the piece that's being copied, your DNA polymerase runs along and hits it. It'll kick off that fluorescent group but leave everything else connected, and then run along and continue the reaction. When it kicks off that fluorescent group is free to react and starts to glow. And this will happen over and over and over again. And so gradually it'll start to become brighter and very clearly, or be glowing whatever color you started with. One of the advantages of it is you can actually have multiple different colors present in your solution. And so if you can diagnose different problems, if different things are present or not present, just by running the reaction. You don't have to run later systems. You don't have to run a gel afterwards to determine what's going on. You can just run your reaction, observe, it's glowing blue. This is what's present, we're ready to go. Or it's glowing green, this is what's present, we're ready to go. So that's the huge advantage you get with the Tachman probe. Both work. Tachman probe is really, really useful. Cyber Green is it's an improvement over normal, just a plain traditional PCR, but it's not quite at the same level as the Tachman probe. Got any questions? Free to let me know, and then these are my sources here. All right. Thank you.